Okay, hi everyone. Thanks so much for being here. I'm Marielle Villeray. I'm the Program Director for Academic Initiatives and Strategic Innovation here at the Graduate Center, the Director for the Lifelong Peer Learning Program, or LP Squared, and co-chair of the Reactivation Committee for the campus. Today, we're so pleased to introduce Barry Levine, a member of LP Squared, and his wife, Shirley Levine, both retired medical doctors, to give a presentation to the full Graduate Center community. They're going to cover today the background of past coronaviruses and the present pandemic, the development, mechanism, side effects, and efficacy of mRNA vaccines, the present status nationally and internationally, and the future, all within the hour. We'll then call up the questions sent in advance for the Q&A. And if you have questions that are percolating throughout the talk, you can enter those via the Q&A tool at the bottom register of your Zoom window, and we'll bring those in. Before we get started, I just want to say thank you to our speakers and to my colleagues in the Office of the President and Communications and Marketing who helped plan today's webinar. I'll hand it off to the doctors and you're good to go. Thank you.
Okay. Thank you, Marielle. We're very happy to be here. Shirley and I are both, we've been retired for many years now. We were both academic physicians, Shirley as a hematologist and I as a surgeon. Which meant that we took care of very sick people in medical schools, taught residents and students, and also ran basic science research labs that were nationally funded and presented our data both at meetings and in papers. We got interested back in all of this when it started, when the pandemic started, and it sort of revved up our academic minds again, which had been a little bit to sleep over the years since we had retired. This was the biggest thing since the 1918 pandemic, even bigger in numbers worldwide than the polio epidemic for when we were children. Secondly, we had a family. We had 3 kids and 8 grandchildren in the New York and DC areas who were at risk from this, and we were interested in finding out all the data to keep them safe. And finally, because we were in our late 70s, we were in the target population that was dying of this disease more than anybody else. And as I mentioned to Marielle earlier, when I first started thinking about this, it brought to mind a quote by Samuel Johnson that's attributed to him that said that nothing concentrates the mind like an impending hanging. And I felt that we were the potential hangees and we better find out about it. What we'll talk about today is not going to be a comprehensive covering of everything there is to know. That would take many lectures over a couple of weeks to get to. As Marielle told you, we're going to cover specific topics and later open it up for questions. First of all, the history of coronaviruses. It's fairly recent. There are hundreds of coronaviruses that are known out there. It's a large family and it exists almost entirely in animals, mostly camels, bats, cats, and pigs. All of the coronaviruses have spikes on their surfaces which lock them onto host cells on a specific receptor that allow them to attack the cells. Of all of these hundreds of coronaviruses, there are only 7 that are known to have shown a transfer from animals to human beings. 4 of them only cause mild upper respiratory disease, sort of like a mild to middling cold. However, there have been 3 that have caused significant disease. The first one was called SARS, which is serious acute respiratory syndrome. Some of you may have heard about it. It appeared in 2003 in Asia, and there were about 8,000 patients who were affected. It had about a 10% mortality and then disappeared spontaneously. Then there was MERS, MERS, Middle Eastern Respiratory Syndrome. This appeared in 2012 on the Arabian Peninsula, and there were only about 2,500 cases, but this had a 35% mortality, much higher mortality, but of lower incidence. And they proved that it was a transfer from camels to human beings. It also spontaneously disappeared. So even though there were some efforts into looking into making a vaccine for these diseases, it never came to fruition because a vaccine wasn't needed. The diseases just disappeared. And that brings us to COVID-19, our present pandemic. It is said to have appeared first in China in 2019. And the worldwide pandemic was pronounced in March of 2020. It has up to today affected approximately 181 million people worldwide, with about 4 million deaths, for a mortality rate somewhere between 1.5% and 2%, which is also about the mortality rate within our own country as well. So that's where we are now. And the next topic that my wife will talk about is the development mechanism, side effects, and the efficacy of the mRNA viruses, which are truly the revolutionary part of this story.
Back in the early part of 2020, when we started to hear about the fact that there was a new coronavirus, I really remembered the first SARS and MERS, and at that time, we wondered whether or not it was ever going to get to the United States, and it really didn't. There were some vaccines that were started, but really didn't get finished because the virus didn't appear here. And then suddenly, it became clear that it was spreading faster than we could ever imagine, and that it was going to be here in the United States. And I remember distinctly knowing that we had our first case on the East Coast on right before March 1st. And then the next day was the lawyer from New Rochelle who had no travel history. And at that time, this country was only really doing testing on people who had been to China, and no other tests were available. And silently, this virus was spreading through the community without us knowing how much of the community was already affected. At that time, our son, who is a physician and who's at NIH, started to talk to me about messenger RNA vaccines. And I was asked, what is a messenger RNA vaccine by the Graduate Center? And apologies to any molecular biologists who are here, but I'm going to try to give you just a synopsis of what captured me about the possibility that we were going to have this new type of vaccine. And the vaccines themselves were the first that were going to be produced as a specific vaccine in the United States or in the world. But this is not new technology. People had been working on mRNA type vaccines or even cancer treatments. And probably it was 10 to almost 20 years where some of this was already being done. So while it said this is brand new and this is so fast and how can we possibly accept it, that really wasn't the pathway. And as a matter of fact, my son was telling me that there was a wonderful young scientist at NIH who had joined the vaccine group, and when asked what it was that she wanted to study, she said, well, nobody here is studying coronaviruses, so I want to do coronaviruses. And she learned everything that she could about coronaviruses. So she was in the perfect position for when it was proposed, actually by one of the leaders at Moderna, which was a very new company and had been doing mainly cancer vaccines, because the vaccine that they developed for Zika could never be tested because there weren't enough patients around in order to be able to test it. And so then I became extremely interested in following this process. So what is messenger RNA? Most of you know that our DNA is our repository for every gene that makes us, all of us. And it's a double strand and contained in 22 chromosomes plus the sex chromosomes. And in order to make proteins, small pieces of the DNA are incorporated into a messenger RNA model. And the mRNA then leaves the nucleus and goes into the cytoplasm, and it is the code for developing proteins. And what became clear to the group at NIH, and this was Dr. Fauci's institute, and specifically Barney Graham's Vaccine Institute, and the young scientist named Kizzmekia Corbett, who actually were able to develop the mRNA that was going to make spike protein. And as soon as they started on this project, they really knew enough already about how important the spike protein was to any coronavirus. And it was the important part of the virus which is an RNA virus that attaches itself to cells. And specifically in us, it attaches itself to a receptor known as the ACE receptor. And all of that was pretty well known. The Chinese back in December had actually released the genetic code for the mRNA coronavirus. And they were able to use that code in order to, number one, construct an mRNA that was directed against the spike protein on the coronavirus, and then to stabilize that protein, and then stabilize the mRNA, which is a very rapidly developing and then disintegrating molecule that keeps all of our bodies humming along. They were able to determine exactly what the mRNA for the spike protein should look like. They stabilized it. In order to get it into the cells, they coated it with a lipid layer, because otherwise there was no way to get the mRNA into the cells. And that was all done within 6 weeks. From the time that it was announced that this was now going to be a major infection, the mRNA vaccine antigen, which would then be given to us as messenger RNA, and that code would be translated in the cytoplasm of our muscle cells, specifically the deltoid muscle cells, and would make spike protein. So instead of giving us spike protein, they were giving us the tiny little message that allowed our bodies to make our own spike protein. And that was the foreign invader that stimulated the immune system. So hopefully this is clear. As I say, I apologize to the molecular biologists out there, but it's my understanding of exactly how this got going. Once a vaccine is planned, there are a number of steps, and I think it's worthwhile to just very briefly mention this. Usually at that point, whatever the antigen is for developing the vaccine, and in this case, it was going to be mRNA developing into spike protein, they need to be able to do some testing in animals in order to determine whether or not there's an immune response and also safety. And a number of different types of animals and even cell cultures are used in order to get through the animal phase. If it looks promising, they then go on to phases 1, 2, and 3 of clinical trials. And these are well established by our government. They've been in place for years and years and years. All drugs and vaccines go through this particular testing process. And for the phase 1, what they're really trying to do is to determine what dose has to be given in order to get a good immune response. But more important than that is safety. And these are very small studies with human volunteers, usually in the 10s to maybe 50 volunteers in order to do those types of studies. And then phase 2 is actually giving it to both, usually both to volunteers as a vaccine and a group that are the placebo control group, which gets saline. And that allows them to determine whether or not number one, the dose is good, and then the efficacy, protecting them against just the virus that they come into contact with their daily lives. And usually those studies are 100 to several hundred volunteers going through phase 2 studies. And then comes phase 3. And most of you know that the results that come out first from these vaccine companies are the phase 3 results. And one of the things that the FDA did, which I thought was amazing, was they established the criteria for what would constitute a good phase 3 trial. And they determined that there needed to be at least 30,000 volunteers And these could be broken up into 1-to-1 treated and placebo groups, or 2 in the vaccine group to 1 in the placebo group, but there needed to be 30,000. And in addition, they really could not apply for an emergency use authorization until 50% of all of the volunteers had already been vaccinated for more than 2 months. Now, with most vaccines, it is said that it takes a minimum of 6 years to make a vaccine. So why did we get a vaccine in less than a year? Clearly, I think it was the Operation Warp Speed, which I'm sure that all of the vaccinologists at NIH and at the FDA and at the CDC were in favor of, because it looked like this was going to be a terrible epidemic. And how fast could they move this along? Usually drug companies come up with their idea is to create a vaccine, and then they do phase 1, and then they have to analyze all of that data slowly and methodically, and most importantly, raise money in order to be able to carry out a phase 2 clinical trial. And then the same thing happens again with phase 3. So it takes a long time to get through all of these trials. And after each trial, they have to submit all of their information in order to be able to go ahead, because the companies are assigned advisors, scientific advisors, to work with them on most of these stages. And so one of the things that made this a fast vaccine development project, which people were complaining about, this was too fast, how could we trust it, et cetera. So that was, I think, how we got really to where we were as quickly as possible. The 2 mRNA vaccines, Pfizer and Moderna, both turned out to be incredibly efficacious, with vaccine efficacy of 94.5% of patients. And it was symptomatic disease that was the endpoint for the vaccine studies. Also, there was demonstration that it prevented severe illness and reduced deaths significantly. So these were very good vaccines, and they were able to come on the market quickly because part of the government funding allowed the companies to start making the vaccine long before they were ready for application to the FDA for an emergency use authorization. So it did not take a year in order to get the manufacturing up to scale. And that was the part that was of great concern. So that's really the history and the development of the mRNA vaccines. And I've already mentioned the mechanism. Side effects were really very limited. Reactogenicity, which is what happens when they give you the shot in the arm and what happens at the site of the shot. People had pain there, they had some redness there, they may have had some swelling there. And other side effects included things that we're used to feeling for some of the influenza vaccines, were a little bit of muscle aches, a little bit of joint aches perhaps, but the safety profile was actually quite good. And the same was true for both of the vaccines. They had very similar safety profiles, and we were able to move ahead. I'm only going to make one comment about the second type of vaccine, viral vector vaccines. That was a DNA portion of the spike protein code that was used for that. And in order to get the DNA into our muscle cells and allow them to actually be a foreign antigen that would stimulate the immune response, they attached the DNA vaccine to an adenovirus, a completely safe, not able to reproduce adenovirus. And so that's why they're called viral vector vaccines. So instead of coating the messenger RNA with lipid, they attached an adenovirus, which easily goes into our cells in order to be able to make the vaccine. In both instances, I think it's important to point out that neither the mRNA nor the DNA vaccine ever leaves our cytoplasm. And these molecules are actually scheduled to be destroyed very quickly in normal operation of messenger RNA. So what else can we really say about the safety? And here I need to go a little bit more into depth because these were such safe vaccines that all we could really say initially was in 30,000 in the case of Moderna, 44,000 with Pfizer, 44,000 volunteers for the J&J vaccines. Really no significant adverse effects had been noted. And the CDC and FDA have an incredibly complex system so that they get regular information from every single, there are like 9 hospital districts around the country for patients. There's a system whereby people themselves can fill out a form of something that they were feeling that they wanted to be able to have the government know about. And this particular system really was collecting almost nothing in terms of adverse effects. And then with the Johnson & Johnson vaccine, they started to notice what was a few more unusual venous clots than they had ever seen before. And the question at that point always is this in the percentage that is seen in the normal population, or has it exceeded those number of cases that are seen on a yearly basis or some portion of time as a more than what would be seen than the normal population? And they finally had, and I've actually attended online most of these long, day-long meetings to be able to see what it is that they're doing. And for that particular discussion, they were really reasonably sure that this was a very, very rare side effect, but it was occurring in slightly more than the average population. And they thought it might be related to the vaccine itself. But interestingly, when they look at how many of these cases actually occurred just when people got COVID, that was way more. So way more of these venous clots in the COVID population than in the vaccinated population. And that really became a safety trigger that they had to look at more carefully. They were then able to figure out sort of why it was happening and how to diagnose it. And it is decreased platelets plus a clot plus a positive test for a platelet factor, a platelet substance, a platelet protein known as platelet factor 4. And really the most important thing that they discovered was that these patients should not be treated with heparin, which is the common anticoagulant for clots that have formed very recently. And for this, they need to use non-heparin anticoagulants. And really there has been very little discussed about these clotting events since they figured out what it was, how to diagnose it, and how to treat it properly. The second set of adverse effects were just more recent, and these are with both of the mRNA vaccines, and that is myocarditis, which is an inflammation of heart muscle, and pericarditis, which is also an inflammation of the covering of the heart. And these are, both of these, myocarditis and pericarditis, are very common with viral infections, a whole host of viral infections. But this was happening within a week and usually within 4 days after somebody was given their second dose of vaccine. And so they went ahead and decided that this was something that needed to be investigated for the same reason. And the advisory group to the CDC and the FDA looked at how many cases there were and what all of the information that they were getting about the myocarditis and pericarditis. The clotting problem was seen primarily in women and relatively young women. The myocarditis pericarditis was being seen in males, and again, young. The myocarditis pericarditis was very mild, unlike what happens with COVID itself or with some of the other viruses. And it was cleared with just things like Motrin, other non-steroidal drugs, and almost universally, everybody recovered within a week's period of time. And so these are the two adverse effects that have been described, and they would never have been picked up in phase 3 trials because of the time that was, the number of people that were in those phase 3 trials. Last, a little bit about variants. The more the virus replicates, the more mutations there would be. All RNA vaccines, or all RNA viruses replicate at different speeds. And we started to see variants being developed first in the UK, then South Africa, then in Brazil. And most recently, the variant of concern is the Delta variant. And so far, all of the vaccines have been able to demonstrate both in the laboratory as well as now in patients themselves, where they look at for breakthrough cases after vaccination, that these vaccines that we have are handling these variants, and now specifically the Delta variant, which is the one that developed in India. It has an increased transmissibility, which is why it took over the UK variant that was in the United States very quickly. And in the United States, it is now spreading. There's about 20% of the viruses that are being typed from the current cases. And it will continue to spread. There's no doubt about it. There are 3 ways that mutations can affect something like the coronavirus. And it can be with increased transmissibility, and both UK and the Delta Indian variants have increased transmissibility. And the the Delta virus is 60% more transmissible than the UK variant. So we will see our country, as the Delta increases, the UK or the Alpha variant is decreasing. And all we can say at this point is that it's going to be the vaccines that save us, that stop the pandemic. And we are hopeful that our current vaccines will provide long-lasting and durable protection, but we don't know that because more variants, there's so much virus around the world that more variants are bound to occur. And the other 2 ways that it can affect us and control the pandemic is through increased severity, which we don't think that we've seen in most of these, or making it not recognizable by our vaccines. And so far, the only thing that has interfered with are the monoclonal antibodies that are sometimes used for treatment of coronavirus. There's a new one, and we can talk about more of this in our discussion, but as of yesterday, there's one wonderful little piece of news. In St. Louis at WashU, they've been studying the immune response in a small number of patients that have been vaccinated. And while after vaccination, antibody levels rise and then they slowly fall, and you hope they don't fall too fast. But in addition, there are 2 other components of the immune response. One of them are B cells, which make antibodies, and the other are T cells, which have long-lasting memory. And what they're finding in studies that they've done in a limited small number of people is that they've got all of the evidence that there's a good memory for this vaccine against the coronavirus. So very preliminary, but it's starting to look as if perhaps our duration is not going to be less than a year or just a year. Perhaps the signs that they're seeing, and they've even looked at lymph nodes to be able to look at exactly whether or not they have the right long-lasting cells that are appearing there. It might be much longer. So that's our moment of hope to end the information portion of our discussion today.
Okay. What about what's going on? Where are we now, nationally and internationally? Well, nationally, as I like to say about everything in life, there's good news and there's bad news. The good news is that we are definitely vaccinating people fairly rapidly over the last several months here. And right now we have a little over half, about a half the population that's totally vaccinated, and a little more than that that has at least one shot. However, you have to remember that no child under 12 has yet been vaccinated. And then we have the vaccine hesitancy, and that's the bad news, in the fact that we are really dropping off in our rate of vaccination as the vaccine hesitancy comes to the fore. And it's gaining strength, if you will, this hesitancy. And if you follow state by state, there are 26 states now that have rising rates of occurrence of positive testing and decreasing, concomitant decreasing rates of vaccination. 26 states, mostly in the South and in the Midwest. But that is a very bad sign. We have plenty of vaccine to go around. So that's shouldn't be the problem. You should be able to walk in almost any place and get a vaccination. Also, we have no vaccines so far for people under 12. They're coming with 6 to 12, I think, fairly soon, within months. But for less than 6 years of age, I think that we're going to be waiting to at least into next year for that as well. Now, what does that mean? That means in the United States that this disease, we will not reach what the so-called herd immunity that everybody talks about, where you don't have to worry about vaccinating more people because you have a large enough percentage of the population, which is usually seen as somewhere around 80, 85%, that they cover each other and the disease can't come out unless the virus changes amazingly. If we're not going to get there, what does that mean? That means that we have an endemic virus. Well, what does an endemic virus mean? Well, our flu, think of it as the flu that we see every year. The flu comes every year. It's slightly different because each flu we get is slightly different than you would here. But about 30,000 in a good year and 60,000 people in a bad year die of the flu every year in this country. Well, since the coronavirus, COVID-19, has a higher mortality rate than the flu has, if this becomes endemic, and we see it every year, we're going to probably get between 120 and 150,000 people dying every year. Right now, at the rate we have, although we have decreased our rate of caseload and a new caseload and our rate of mortality in this country over what it was at its peak, we are still going to lose somewhere between 9 and 10,000 people a month, which is unacceptable. Unacceptable. We will be very shortly, that means, well past the total number of deaths from the 1918 pandemic, which was 675,000. We're at 605,000 right now. And we'll get to 675,000 soon enough. So that's the bad news. The other bad news in our country is what it's done to affect our overall lifespan in this country. It was rare for this country to have a decrease in lifespan year over year. We were always going up and up and up. However, in the last couple of years before COVID, it had actually started to go down a little bit, about 0.1 year each year, which is not much to worry about, but it was a change of direction. Well, in the last year to year and a half, there's been a decrease in the average lifespan in the United States of 1.9 years. That's never been seen in a long, long time. And in the white population, it was 1.4 years, but it really was quite awful in our minority population. The Black population had a decrease of 3.2 years in longevity, and the Hispanic population had a decrease of 3.9 years in their longevity. This is going to take a long time to reverse, to get us back to zero again, and then start up and increasing our lifespan. And that's also the bad news. Now, internationally, there's even less good news, because although we're awash in vaccine in this country, we are not awash in vaccine internationally. We're way behind. Although we are giving away millions of doses, that's not even beginning to fill the bottom of the bucket that we need to fill. Remember, there are, over 3.5 billion people out there right now, and we need a lot of vaccine to cover them. The other problem is that there are not a lot of public health measures being put forward in several of these countries. So, for instance, in Latin America, in Brazil is like nobody's watching. I mean, it's uncontrolled. It's virtually, when you have a president who didn't take the vaccine in Brazil and who said that he didn't believe in the disease, there's part of the problem. South Africa is another problem. Even West Germany now is relocking down. Bangladesh is locking down. Australia is locking down again. So we are far from out of the woods. And remember, the United States is not an isolated entity in this disease spread. We cannot isolate ourselves from the rest of the world. Even if we so-called conquer the disease in this country, we cannot stop it from re-entering, because worldwide our population moves and it carries the disease, and the virus is an opportunistic infection that will find a way in. So the future depends on us getting a workable vaccine out to the whole world so that we can inoculate people. Because unless we do, we are going to see an endemic problem in the world where we have even a low level of the disease, but a low level of this disease will cause hundreds and hundreds of thousands of excess deaths every year in the world, and at least 120,000 excess deaths a year in the United States over what we're seeing now. And on that pessimistic note, I'll end and open us up for questions. Marielle?
Thank you both. So we accepted questions in advance of today's webinar. So I'm just going to review some that definitely you touched on, but maybe we could go a little bit deeper on certain topics. So here's one. If someone has been vaccinated but is exposed to people who have active COVID-19 symptoms, what are the risks? Can a vaccinated person still spread or transfer COVID-19 from one person to another?
People have been worried about the fact that there could be transmission from a vaccinated person because of low levels of virus in their upper respiratory system. And it looks as if that probably is not going to turn out to be a major way of passing on the virus. But I'd say that from most of the discussions that I've heard, yes, they don't think that it's going to be a problem, but they don't have the absolute proof that there are no cases that are being transmitted that way. So, yeah. I think the whole issue about when you wear masks and when you don't wear masks would take the rest of this hour and maybe the next hour. And so I think that we should just leave it with the CDC recommendations, which are that if you're vaccinated, you can go outside, you don't have to wear a mask. If you're inside, it really depends on where you are, what the risk level is, how much virus is present in that particular community. If everybody's vaccinated and you're inside, you're probably okay. I mean, those are sort of the general guidelines. But I think that as I look through some of the online discussion groups with the epidemiologists, there are hours of questions about very specific things. And especially now that people are parents who are vaccinated or grandparents who are vaccinated and being around kids who are below 12. And can you be bringing them a little bit of transmitted virus, or could they be going to school, going to sporting events, et cetera, and bringing virus home? And there definitely are breakthrough cases. And I think that that's a very important concept in everything that we talk about. People who are vaccinated can still get COVID. There's no question about it. You know that it's 95% efficacy in just the original virus for the mRNA vaccines. It's less for the J&J. And so you have to really know, there have to be studies of the breakthrough cases, figure out whether it's due to variants and how many of them are due to variants, and then calculate, is that the 5% that weren't going to get a good response to the vaccine? And all of this is being done constantly. And we just have to hope that New York just started doing genomic sequencing in February. They had done none before that. Any information that we had about the genomic sequence was coming from the UK, who were all over it. They were sequencing everything. And now it's picked up, and they're doing now about 8% of the viruses that are from cases here in New York City, not the state, but the city. And the Department of Health lab here is sequencing them, which is laborious. It takes an enormous amount of time to sequence a virus that's been isolated from someone who they collected a sample from. And they're publishing the data, but it's not something that's really looked at that much. As I mentioned, the last data on Delta virus here is 22% that's now been isolated. There was none in April. And so April to now, we've gone from zero several weeks in April to 22%.
Now, the thing that should be, I think, underlined, however, is yes, there are breakthrough cases for vaccinated people. We know it's pretty low. We don't know how, if we included people who are asymptomatic. You have to test to find out the people who are asymptomatic, and we're not doing enough testing for that to know what the real number may be. But we know that if you get COVID and you've been vaccinated, you're not going to an ICU and you're not going to die. And that's the biggest thing that anybody has to know about being vaccinated. And that's the difference between being vaccinated and not being vaccinated. When you get COVID and you're not vaccinated, overall, a 1-point-something to 2% chance in the United States that you're going to die. If you're vaccinated, you're not going to die.
For people who were afraid that this vaccine was made too fast, as I mentioned, this is technology that has been going on for a long time. And there are now 150 million people who've been vaccinated with the mRNA vaccines in this country. And there have been just those 2 little instances where there's an adverse effect that is so rare. We're talking about a couple of cases per million doses given. And one of the things that is really on the airwaves now is it's going to decrease my fertility. There is absolutely no evidence that that has happened, that that could happen. And some of these fears are just irrational. So it wasn't made too fast. It wasn't made without all of the precautions that they always take. And the government has advisory committees, scientific advisory committees of faculty in all aspects of this field that meet with the committees and are the ones that make the recommendations on acceptance or not acceptance. Is it connected? Is it not connected? It is one of the most incredible processes I've ever sat through. And I've sat through all of them.
Yes, you have.
All of them.
I don't, she does.
Last week was myocarditis and boosters. And the final decision from the advisory committee after hearing a 2-hour presentation was, we don't have enough data to know whether we need boosters yet. But something that I failed to mention, all 3 companies already are making booster vaccines, and they will modify them as necessary. But because the mRNA vaccines can be made so quickly and the vector viral so quickly, we will be ready. The issue is, will we need them? And they have so many things in place to make that decision of whether we need them and when we will need them. And I think that the newest thing that's coming out on how much memory our bodies have
for this.
now, after months, it was 8 months after the vaccination that they were doing these studies by just taking little syringes full of cells from the lymph nodes in the arm and looking at the fact that there are the memory cells that are necessary, that are there. And they look like they're very active and continuing. And that, I think that's one of the best things that it has to be, like every experiment in medicine or science, it has to be replicated by other laboratories, and the results make sense. This was published in Nature, which is one of the best journals, and peer reviewed in order to get to the point where it's being published, and it will come out after it's proofread, which is where it stands right now. Yeah, soon. So maybe we're in good shape.
I don't know how many people are in the audience who are either wanting to be pregnant or are pregnant or have a spouse who's pregnant. I wanted to come back to that for a minute, because the whole idea of these vaccines having an adverse effect on pregnancy was an idle thought off the wall by one person that gained currency somehow in the social media of the United States.
Yeah, about the fertility question specifically.
And it's just not there. There is no data. And in fact, the data, now that we've piled up, there are lots of pregnant ladies who have gotten vaccinated now, or women who were vaccinated and then got pregnant. And the data would say that they're fine.
In addition, the effect of COVID on a pregnant woman is much worse than it is on a non-pregnant woman. And they're recommending vaccination for that alone. Less premature babies, less stillbirths, less complications with the mother. And in addition, the antibodies to the virus are passed along in the placenta and breast milk. And so it's recommended that every pregnant woman discuss the vaccination if they already haven't gotten one with their obstetrician, but that it favors vaccination as opposed to trying to do it without protection, because it's a more serious process that happens if you're not vaccinated and you're pregnant. More complications.
You touched on a lot in that, but I wanted to go back to the comment about children getting vaccinated. And a question has just come in. Why are the current vaccines not suitable for children under 12? And what about the 12-year-old body changes the ability to accept or tolerate the vaccines?
Children are never included in the original phase 3 clinical trials, ever. Neither are pregnant women, because until they actually have real-world experience with these vaccines, they're very afraid of things that might be missed. With kids, as they go down from 12 to 6 months, and that's 3 age spans that they will be testing, and all of the companies have already started that. The first thing they have to do is find a dose. What's the dose? They can't use the adult dose, although that gets used in the 12-year-olds and up to 18-year-olds, and they handled it fine. But they have to find the dose, and the dose, they have to start low. And their goal, their endpoint, is to have as much vaccine efficacy as in the 18 to 25-year-old group from the clinical trial. So they want to know that it's as effective as anything that they have out there, but it's going to take a reduced dose. Normal, and that's called immunobridging, that they go down in age until they come up with the right dose, and then the efficacy issue is really paramount. And so they're going to go slowly, although they're starting to enroll kids in the different age groups so that they're ready to be able to do it a little bit more quickly. One of the things that's said all the time is that kids have a very low risk. But as one of my favorite podcasters says, but it's not no risk. They have risk. And so they still have to be protected. And they will still be wearing masks, and they should be wearing masks, especially if they're among people who are not vaccinated. one of the places where there was an enormous spread of the vaccine were in sports, school sports.
Birthday parties.
Birthday parties. That's the latest one, is that most of the spread now into kids is at birthday parties. So I think we're all very worried about kids, but they do have a lower risk. One of the things that is most worrisome is that, yes, they can get COVID, but they can also get sort of a hyperimmune response after they develop COVID, which is called multi-system inflammatory syndrome, MIS-C, for the childhood variety, because it also can occur in adults when they have a hyperactive immune response to the infection. And so there's a lot of reasons that we don't want them to be getting COVID or to be in that small group that can go on to develop MIS-C. But they will do this safely. And that's the only thing that we can really ask is be safe. Don't give our kids things that are an issue. They know what some of the side effects are. And unfortunately, the myocarditis turns out to be in relatively young males, which males more commonly get myocarditis anyway from viral infections, not just COVID. The other thing that they found was that in the Big Ten athletes, they looked at 1,500 of them who had never been vaccinated, and it was something like 2 to 3% of them had evidence that they had already had COVID, that they didn't even know that they had had it. That's a very different kind of myocarditis, because once they know that they have that, they do no exercise for 6 months. They just want the heart to heal as properly as it can. But in these kids that are getting the myocarditis secondary to the vaccine, it is very short-lived. Days that they have some chest pain, maybe a little bit of shortness of breath with exertion, very little else. And we hope that it will not go down in age, that there will be more, but still the risks versus the benefits were in such that the advisory committee, after hours of going through all of the data that exists, said the benefits are so much greater than the risks of this very, very low incidence of myocarditis with the messenger RNA vaccines. So we have to just wait and see.
Well, and when people say, little kids, they just fly through this, they're not, they don't get it. And when they get it, nothing happens. 400 dead in the United States.
And almost 4,000 with MIS.
With the inflammation thing. So it is not benign. And if we just had a disease that was segregated to kids alone, that adults didn't get, with numbers like that, we'd be pulling our hair out in the United States to find something to treat it with. And we're going to have something to treat this with before. It's called a vaccine.
And for those who are immunocompromised, what is the efficacy of the vaccines for those people, and what are your recommendations for behaviors and habits at this stage of the pandemic?
Variable who responds and who doesn't respond. People that are getting cancer treatments may be not responding. People who have had organ transplants or who are on high doses of immunosuppressives, or people that have immunosuppressive diseases or on medication for autoimmune diseases. It's variable. Some of them have 0 antibodies. One of the points that I should make right at this moment is antibody tests should not be taken by the average person. First of all, there are 2 types of tests. One of them will come back 0 antibodies because it's not measuring the spike antibody. And nobody ever tells you what test you're getting. And so the recommendation is don't bother getting the tests unless you are immunosuppressed or have some reason to be immunosuppressed due to therapy. Don't do it.
And by the way, if you are immunosuppressed and you need to go for a test like that, make sure that you talk with your doctor and say, I want the right kind of antibody test that will measure this.
I need the antibody test to the spike protein.
Right.
And then they probably, if this really is a major consideration, they probably should get a very good antibody test that titers it so that they know if they have low antibody, medium, large, strongly positive. But there definitely are people that have 0 antibody. And I personally know 2 that have 0 antibody. They're now thinking that those people should get a 3rd dose, but that is totally the consideration of the physician, the patient, and what is known about maybe a 3rd dose, boosting them to a level that will be good. And immunosuppressed people know who they are, and they should continue to wear masks and every single other epidemiologic maneuver that we have to prevent spread.
And at the first sign of them having any symptoms of this disease, number 1, get tested, but number 2, get a monoclonal antibody, because that's the only treatment that can help them at that point. That's all we know at this time.
Now, treatment has really suffered.
Yeah, we don't have treatment.
They've tried everything. And so far, the monoclonal antibodies turn out to be one of the best if given early in the disease process. And these are antibodies to a very specific part of the spike protein, usually in a cocktail. So that these are just a single antibody directed at a single spot on the spike. And they put a couple of them together. And convalescent plasma doesn't work. They finally have come out with the data that has, from the beginning, showed that it really didn't work, but people weren't willing to give it up. And convalescent plasma is plasma from one person who had COVID. Monoclonal antibodies are very specific monoclonals that are present, and they've been made, and they are there in high concentration. And the problem is that most of them have to be given IV. And if you do it early, it's an outpatient, and it's a very expensive process. But people are being told, once they get an antibody positive test, if they're still in a situation where they might be able to get monoclonals, ask for it, find out.
So on another topic, someone wrote in that they were vaccinated with Johnson & Johnson in March and asked if they should get an mRNA vaccine as well, and the timing of that, how soon after the initial J&J inoculation.
Heterologous vaccination is using 2 different vaccines. And in the United States, people are, and I'm talking about expert scientists, not us, but they're saying that we really aren't ready to do that without study. I mean, we do clinical trials in order to prove that what we're going to do is not harmful and is efficacious. And Britain is doing it.
They've been doing mix and match all over the place.
And also expanding the time between the first and the second dose, which also may not be something that we would ever do here without very good trials in order to determine it. And in the case of having gotten a J&J, the J&J vaccine is good. I mean, there's really no problem with it. And whoever that person is, I would just recommend that they stay tuned and listen to what's going on in terms of repeating a level or repeating a vaccine. But if they're not immunosuppressed, they also are protected against severe disease and death. So they should consider themselves protected.
Great. And just one final question for you. What are your tips for making sure that the N95 mask that you're buying online is surely going to protect you?
Okay, well, first of all,
we wear homemade masks.
Yeah, well, my wife makes our masks.
Multiple layers.
She's a quilter, and she has quilting fabric, which is very densely woven. So she puts 3 thicknesses and puts pleats in them, so they actually fit your face. But at any rate, yeah, so an N95 mask, they used to be very hard to get. They're not very hard to get now. I just, when actually you mentioned this the other day, I went to both Amazon and Google immediately. They're all over the place. Now, some of them may not be real. Now, on Google, you should just put in, how do I know that my N95 mask is real, or something like that. And you will get something that comes up that has 7 things on it. And in fact, there are a couple of different sites that list these 7 things and how you can make sure that the masks you buy are the real deal. But there are plenty of them out there, and they're not that expensive anymore. They used to be very expensive when you could find them. but they're not. So just go online and look for the 7 things you should look for, and you're going to find them. They'll be there.
I think a more important question is, when would one want an N95 mask? And I don't have an answer for that.
We still have a box of them in the back that we haven't used.
Yeah.
And the cloth masks do just fine for every day
Yeah.
going around town.
Very much so. I think if you're going to be on a long flight, crammed into a airplane cabin, then I would want an N95. And I have friends who also then wear a shield with it as well. Since we don't travel very much, that hasn't been an issue. But we did wear N95s back when the virus was so high and New York was so awful. In March and April, we wore it once every 2 weeks when we went to the senior time at the grocery store.
Right.
That made us feel more comfortable.
And you have to have it fit too. You know, when people used to wear N95 masks in hospitals before all of this, they usually, before they were in a job that required an N95 mask, got fitted so that they knew how to wear it.
It's actually called fit testing.
Yeah.
And what they do is there's a little spray involved, and it's when you can taste and et cetera. And this would not work.
Yeah, beards don't make it.
No, at all. And so if you don't know that it's fitting you absolutely properly,
you might as well use something
I was going to
else.
say it might not be a whole lot better. Surgical masks with the gaps on the side make me crazy. I mean, the masks that I make actually have the elastic that goes right through the side. So it is really tight.
Oh, by the way, any mask that says it's an N95 and has the ear loops on it is a fake.
Well, these are really helpful tips.
Yeah, it has to have the 2 separate elastics, one that goes
Right.
over head, the other around.
And that was how I chose the pattern that I used for all of our masks, was to make it fit like an N95 so that there's no, you can't even figure out there's a nose bridge on it that fits carefully and really tight. When you take it off, you say, they feel good.
Yeah.
Yeah.
Well, thank you so much for the tips and the education on the science background on these vaccines and the encouragement. We really appreciate your time, and thanks to everyone for joining us today.
Well,
Thank you for inviting us.
Yes, thank you. It was our pleasure to do it.
And our brains are a little bit more alive.
Yeah.
Ours too. Thank you, everyone. Take care.
Take care.